One of the things we found is quite useful to do with people is to do a scale of what's called transcription. We can call this a scale of epigenetic health, in other words, the best that your genes could be. So, the scale of transcription really means your ability to reproduce your DNA from the coding strand here to reproduce it into the RNA. So we've got the coding one, which is what you're actually made of, if you like. This is your blueprints, this is your drawings of your building, if you like, which is here. And we know it's a double helix, so we have the coding strand and we have the template strand. The template then reacts with the RNA and forms the exact duplication of the coding strand. So this is called transcription. So your ability to transcribe or change from your DNA, from your blueprint, then into making the new buildings, the new building blocks for your building, is what is the aging process. So does anybody want to volunteer for this? Finn, yes, okay, good. So your scale of transcription. So all I'm going to do, instead of saying a scale of health, I'm going to do a scale of transcription. Scales of health, I've had people up quite high and yet can't walk up the stairs. I'm sure we've all done that. They come up with 95% and yet they still can't really walk up there because they're lying down on the treatment table not doing anything. So, you know, the body's saying, or the brain is saying, well, it's not too bad. But the scale of transcription, let's say your scale of health. Let's do the standard scale of health. 1 to 100, with 100 being the best you've ever been in your life. Right now, you calibrate at 90% or higher, 95%, 100%. So, boy, he's coming in at 95. Fantastic. Scale of transcription. That means the ability for your DNA to reproduce itself into the RNA to make the proteins. Exactly the same as an epigenetic. In other words, if everything was perfect, the best your genes could be, okay? In a perfect world, you know, and the best you can be as opposed to the best you are at the moment. So, right now, out of 100, your scale of transcription, your ability to reproduce your DNA, calibrates at 90% or higher. Oh, calibrates at 70% or higher. At 60% or higher. At 65%, at 70%. Now he's dropped right down to 65, which is probably more realistic as to how he feels. Okay? So he's coming in at 65, which is much lower than the 95 we got him up just treating him on the bench. So now we've got something to really work on epigenetically and to say, right, let's try him out in the process that we did with Jerome, with the different colors, and find out what it is that builds his scale up. Thank you. So with Jerome, I want you to imagine a 7-rung ladder. Okay? So the bottom rung was the important one, wasn't it? The bottom one had the most information, which is why you start at the top and work your way down to find the one that weakens. So you'll only find one that will weaken. Okay? So as you work your way down, that's the one you treat. You stop and you treat, and you find the different eye positions and so on, which will do that. The bottom rung was broken with him. You can't fix the top rung, can you, if you don't fix the bottom. And the top rung was the valine. Okay? We can give him all the valine we want for the repair of the tissue, but we can't if we've got the bottom rung. So that bottom rung is the first one to fix. Sometimes people go through various rungs. Sometimes it's only the top rung which you've got to fix. But if you go through the different rungs, you'll find out which one you start. So always start at the bottom, and then you can build your way up. So once you repair the bottom, you've got a foundation to start building your other stories in the house, right? And each one of those will correspond to a reactive oxygen species. So we looked at challenging with the biophotons. We did all that, we repeated it, and then we looked at each one, or you can look at each one through the eyes into distortion. Now, I don't want to spend time on that, because we're all familiar with the different eye positions. The one I want to just very quickly show you, and then I'll be more or less finished, is two things that we found out. On the nutrition side. The first one was about selenium. Now I bracketed the word phosphate here because this is not selenium phosphate, because you can't buy selenium phosphate. You can buy selenium and you can buy ATP, which donates the phosphate. So that's why I put it in brackets. So this is not selenium phosphate, it's selenium phosphate in brackets, all right? That's why. So Harper's biochemistry, biosynthesis of selenoproteins, that's where selenium works, requires a requires a sulfur amino acid like cysteine, cysteine, methionine, glutathione, etc., plus selenate. So sodium selenate makes an ideal substrate here, plus ATP, plus water. So this is important things that when you use this compound, which you will enormously, is you must open or instruct the patients to open the capsule up, put them into water. You don't have to stir it, but you can stir it for speed. It fully dissolves. Sometimes you've got to just leave it there for a few minutes for it to dissolve, but if you just stir it around with a spoon, it dissolves much quicker. Plus a selenium-dependent enzyme, which is called selenophosphate synthetase, which actually requires selenium to activate the enzyme. So it's interesting, by giving selenium, you're activating the enzyme to do it. So the major components here is selenate, sodium selenate, and ATP, and water, which is what you're going to mix it for. So we put in 100 micrograms. So selenium, you're talking micrograms because it's a real trace element. And adenylyl triphosphate, 100 milligrams, just to make sure the damn stuff works. Okay, so ATP is what you make inside your body there. So the ATP is to donate the phosphate group to selenium to make selenium phosphate. Open the capsules up and dissolve in water before swallowing. We do them as small capsules because some people only need one capsule. Okay? Now the RDA of selenium is about 60 micrograms. The safe level is considered to be 800 or 1,000. Okay, it's a bit dubious, and that's just as a conservative level. But most people are happy 400 to 500. They don't like sort of what books say, well, we halve the highest level to be on the safe side. So we do it in 100 micrograms for that reason. Most of my patients are 4 or 5 100 micrograms, 4 or 5 capsules they need for about, again, 2 weeks, and then the dose halves. So they've got to flood the body in to get it in. For a long time, I've used selenium as selenium cysteine. We've used sodium selenate as a liquid. We've used selenium methionine, selenium picolinate, selenium yeasts, you know, all these sort of things. They hardly ever show. Occasionally they show. But this stuff shows very, very commonly. It's amazing. That's why you've got a sample of it for testing purposes. There are now 25 selenoprotein enzymes have been identified. But the metabolic function has only been identified for about half of them. Okay? So there's a lot of selenium enzyme proteins out there, but we don't know what half of them are for. Okay? But there's more and more all the time being discovered why selenium is such an important nutrient. We were at the dentist the other day, and lo and behold, the dentist was using a selenium liquid because it's very important to chelate out. It bonds onto mercury when it's released in the system with the dental amalgam filling. So they give it that to stop it from getting into the system because it forms an insoluble compound. So the two main selenium-dependent enzymes are the thyroid deiodinase, which is T4 to T3, which you talked about this morning, and glutathione peroxidase. So this is where the T4 to T3 works. 35% of T4 is deiodinase to T3. 45% of deiodinase goes to reverse T3, which is like a blank, and the rest is conjugated out through the liver. Our experience is that most people have a conversion problem, T4 to T3. They feel the cold, okay, and they have all the symptoms of hypothyroid. They have the blood test done of T4, and sometimes they even have a T3, and the results come back normal. Okay? Because you can't measure T3 in the cell, which is where it works and where it's made. What T3 is in the blood is produced by the thyroid gland, and it's a relatively small percentage. Most is T4, which converts inside the cell. So it's impossible to really do a determinant of T3. Apart from the body temperature, the metabolic rate and the muscle test is a much better way. But the second important enzyme with these people is glutathione peroxidase, because if you're short of selenium, you're short of T3, but you're short of glutathione peroxidase, which recycles glutathione to detox. These people have toxicity problems, enormous detoxification problems. They're chemically sensitive. Okay? Now you begin to think, I've got patients like that, I'm like that. I feel cold, I'm sluggish, there's weight gain, and I'm toxic. And, you know, then suddenly you think, oh, wow, you know, I'm chemically sensitive to a whole range of chemicals. Remember, glutathione is the major detoxifier inside the cell, which includes the neuron. Here's the glutathione peroxidase enzymes: cellular, plasma, phospholipid, gastrointestinal, and olfactory. 5 different types of glutathione. They're all selenium dependent. So we'll whip through this. So, because I want to get to fat-soluble toxins are primarily metabolized in phase 1, phase 2 liver detoxification. The most powerful component of this system is glutathione. Fat-soluble toxins can completely eradicate total glutathione levels. Fat-soluble toxins can completely eradicate total glutathione levels. So you have a fat-soluble toxin that has to go through phase 1 and 2. It can wipe the glutathione out completely. When glutathione is depleted, all of the thiols, that's the sulfur brain code, become depleted, including SAM, cysteine, methionine, cysteine, etc. Toxin exposure through glutathione depletion collapses methylation in the SAM pathway. Cysteine is the rate-limiting step in the synthesis of glutathione. It is freely converted into glutathione, be it during weight loss or other fat-soluble toxin exposure in the course of a normal day-to-day activity. The glutathione may become completely depleted. There are hundreds of articles which link methylation collapse to, especially in cancer. So Jill's going to be describing this, the individual ones like the methionine and the cysteine. So glutathione, most powerful antioxidant, most powerful detoxifier conjugator here. It's a modulator of leukotrienes and prostaglandins 2 series. So it inhibits lipoxygenase enzyme, which is what shunts arachidonic acid down into making leukotrienes instead of prostaglandins 2. These are people who get a lot of inflammation, don't they? So leukotrienes is the most powerful pro-inflammatory chemical known. Okay? Let's just think about it. You've got someone with a sluggish metabolism, they're full of toxins, they've got high reactive oxygen species, antioxidants, and they've got pain. Isn't that most of our patients? And you think, what's the common theme through here? So the common theme could be simply selenium deficiency. Why? Where's the selenium? Selenium's in the soil, isn't it? And we've intensively farmed the soil. Okay? The major intake of selenium is wheat. Okay? But we've grown wheat on the field year after year after year after year. There's only one place really in Great Britain where selenium is still okay, because it's reclaimed land, which is East Anglia, which happens to be the area where people live the longest in the UK. So it may be significant there. Okay? And because selenium was there present because it was reclaimed land from there. The areas of the world where the selenium level is highest is where people live the longest. Okay? So it's worthwhile studying these. Now, I talked briefly this morning about camel wheat being discovered in the pyramids. And the thing about camel wheat, it was rich in selenium. So the place to grow it is where there's selenium in the soil. And this farmer who was on duty on active military service out there at that particular time took the wheat back to his farm in Montana. And Montana and Dakota, that area around there, is naturally very rich in selenium. So he has the world monopoly on camel wheat. So he has the patent, if you like, on the cereal. Nobody else wanted to know. He was ridiculed for 20 years when he grew this stuff as a crank. It then went into disuse, went out in the 1970s, but his son reinvented it, if you like, and brought it back into the market when people started getting sensitivities to other forms of wheat. And now it's become very popular. And you know the biggest buyer of this in the world is Italy. You go to Italy now, and particularly Sicily and southern Italy, and there's camel pasta, and there's camel pizzas, camel bread, and people who can't tolerate wheat over here can tolerate it over there. And it's because it's a different form of wheat, you see. So glutathione is recycled from selenium, and the really only good source of selenium as a phosphate group now, naturally, is camel wheat. Okay? Beets watercress, beets Brazil nuts, all the other natural sources of it. So other selenium-dependent enzymes, thioreductase reductase. Jill, do you want to be cutting some bread, or shall I do that? Okay, I'll do it while you're talking then. Okay, thioreductase reductase participates in the regeneration of antioxidants. Selenoprotein P is found in plasma. Protein W is found in muscle. Synthetase is the one that actually makes the selenium phosphate compound itself. Methionine sulfoxide reduces the oxidation of methionine. CEP15 is a mammalian protein located in the endoplasmic reticulum, may have a role in cancer prevention. Selenoprotein V is expressed exclusively in the testes and is thought to function in spermatogenesis. Selenoproteins is involved in the retrotranslation of most folded proteins. This protein is involved in inflammation. Selenium neutralizes methyl mercury in the central nervous system. So this is one of the best ways of getting methyl mercury out of the central nervous system. Should always be administered with dental amalgam removal, and especially when NAC or cysteine is used to stimulate glutathione. Okay, so that's that one. So start to use the marker. We've got a marker for everybody, and we've got a sheet of paper which basically traces what I've just put on there for you. But when you get patients like that who are tired, toxic, and in pain, which is everybody really. Now the other one I want to show you, which was a surprise to me, was vitamin D. Now, vitamin D. Jill, can you get Jerome's markers back out again? His vitamin C. Jerome, can we borrow you up again? So, vitamin D, we know, comes from the sunshine. Everybody knows that now. On the Simon Mayo program, which I listened to the other night coming home, the homework question was, if you wash your clothes and turn them inside out and put them on the washing line, and then wear them unironed, but turn them around the right way again, do you get the vitamin D? What a question to ask. They needed an expert to actually say, no, this was not the case. But perhaps the answer was that if you rip them, you probably would get more vitamin D that way, you see, in the process. So vitamin D. Cholecalciferol is made from the sun, we know, at about 290 nanometers, so it's well out of the human vision. It's in ultraviolet A, so it's a dangerous level, so you don't want too high a dose. But we build our vitamin D up for the winter, hopefully, now. You'll see that we hydroxylated on the 25 position, so it's called 25-hydroxycholecalciferol. This is the storage form, okay? Remember, we do all these as markers. They're in your kits. If you haven't got them, then this is the kit. You certainly want these markers, because you'll find that vitamin D deficiency is pretty rampant, particularly in our patients, musculoskeletal patients, who have a lot of bone aches and pains and things. So the 25 is inert, just like cholecalciferol. It's stored in the liver, the muscles, and the fat over the winter for us to be able to use during the sun. Now, when we hydroxylate this in the 1 position, so we've already hydroxylated in the 25, you see these enzymes are called cytochrome P. These are cytochrome enzymes. What does that mean? Cytochrome means they're light sensitive. Okay, so these are all stimulated by light. But which light? We know which light in a minute. It's his complementary color light. Okay, so we now hydroxylate in the 1 position. So this is now called the 1,25-hydroxycholecalciferol. And this, for most of the time in Harper's and other biochemistry books, is considered to be the active form. But you can't measure it because it only has a half-life of 3/4 of an hour or so. Okay, so it's very inaccurate. If you measured the level, you'd never find whether the person needed none. So what they do is they measure this. But this one is not often deficient. It is sometimes, but it often doesn't muscle test as being deficient. This one, very rarely. I never found using this activated form. You know, once in a blue moon, wasn't it, Sam? Because you were the same as me. You found the 125 actually strengthened anyway. But this one was considered to be inert up to fairly recently. It's called the 2425, which is then hydroxylated in the 24 position and the 25 position. And this is the one which has shown to have receptors in the prostate, the testes, brain, skin, breast, lungs, colon, heart, immune cells. This one is the one we're interested in with the immune modulation, prevention of autoimmune diseases like MS, RA, except type 1 diabetes, prevents infections, regulates cell growth, improves mental health, regulation of blood pressure, and anti-inflammatory. That's what it's supposed to do, all right? But it doesn't always work because there's a third active form, and this is really very new. This is only in journals at the moment. But it's very easy to test for, and because it's a combination of the 125 and the 2425, and it's called 12425, and it's considered to be a metabolite which is excreted by the body. But it is the biggest hormonally active form of vitamin D. It's with the 3-hydroxycurcumin. It's a hormone modulator. And it modulates all the hormones in the body. Not just the steroid hormones, but it modulates the thyroid. And I got into it because it modulates the pineal gland. So it's a modulator of all the hormones. And so when I ran through all the journals and things, and then eventually I was so pleased that I found it validated that it is the hormonally active form of D, which explains the sort of third part of the jigsaw puzzle. So what we're going to do here is, or what I've done here, is we put on the vitamin C and the valine. We ran through all the active colours, didn't we? All right. So now what we're going to do is we're going to put the 380 on him. The dreaded 380 in the clear. Okay. So 380 in the clear now. Remember, he didn't weaken to that to begin with. We're now talking about genotoxic. We're talking about his genes. Okay, we're now getting into the real issues with him because it's his genetic makeup and where there's something genotoxic or a polymorphism in here. Okay, so he has got a problem, and we've all got this. So don't worry at what stage you weaken to the 380. It just means you've got something which has damaged your genes. Could be inherited, could be acquired. All right. Now there's one thing that will sort this out. Give me the different forms of vitamin D. So start with cholecalciferol, which is inert vitamin D that we get from the sunshine. So no good you going out in the sun. Well, you live out in the sun most of the time, so you should get plenty of that. The 25 is the storage form. Okay. Now we do the 1. Okay. This is the 25. So we'll do the 25, which is the storage form. Okay. Now we do the 1. Give me the 125. Okay. And now give me the 24-25. And this is where you would have said, he's fine with his vitamin D, and I would have as well. And I would have sent him packing. Said, no, you don't need vitamin D. But let's put the 2 on together. So now what we've got is this last bit is we're activating one of these 2 pathways to make 124-25. Okay? So now let's see if this works with him. Oh, this works against the 380. That's impressive. This is against a genotoxic substance or a genotoxic effect. Right? So which one is it? So let's take everything off. Okay, so it could be this one or this one. Okay, which is defective. So is it the 24 conversion, which would be the 1, adding the 1 hydroxylation on here? So if this was the case, there's a defect in this enzyme. This would be the defective enzyme, wouldn't it, in his body that he's got a genotoxic effect on. He would weaken to this and strengthen to this, which we know he does. So we'll put the 24-25 on. So we're working just in the clear there, and he's nice and strong. So we do the 125, which is this one. So if he's got a polymorphism or SNP here, he would weaken to that and strengthen to this. We know he strengthens to that. So now let's put the 125 on. Right, so you now know where his polymorphism is. It's here. Because he can't make this. So if he can't make this, he can't regulate his hormones. So his adrenals will be on the blink, his pineal could be on the blink, his testes could be on the blink. And that would be devastating, wouldn't it? So, thyroid, all the hormones, this regulates and balances all the hormones. How do we get this to work? The light therapy works, okay, we know that to some extent, but there is one compound that will do this. One compound, and one compound only. And this is butter. And this butter is unpasteurized butter. Okay? Because pasteurizing destroys the enzyme, the 125, or the 24-25 enzyme here, to convert it. Because they heat it. It's the natural source of vitamin D. Every other books will tell you, isn't it? Butter is the natural source of vitamin D. Have you ever found half a pound or a pound of butter ever strengthen anybody with a vitamin D deficiency? No, it doesn't do it, because it's not the D. Right? Have you ever found margarine would strengthen anybody with vitamin D? No, because it's cholecalciferol they put in. They can't activate it. Nature has given you the enzymes within the milk to actually do this, and these enzymes are often made in the gut of the animal. So we take the milk, which is a source of these enzymes, And that enzyme is then destroyed in the pasteurization process. So you lose the hormonally active actually part of vitamin D. Now it pops, the idea with the 24-25 and the 124 is it puts calcium back into the tissue. The 125, which has got high, will take calcium out of the tissue, but what will put calcium back into the bones is the 124-25 or even the 24-25, which he's got this one, but he's got an excess of this one and not enough of this one, right? Now, it just happens the other thing that will do that is what Jill's going to talk about is glutamate and vitamin K2. K2, yeah? So what is K2 in? Natto, yes. But natto is not a common substance. What's it in? Butter. Well, the books will tell you it's butter. Okay? It's from fermentation in the gut of the cow. It's the bacteria in the cow that make the K2. The K2 is absorbed into the cow. The cow then secretes it in the milk. But milk is 97% water. But butter is 100% fat. And vitamin K and D are fat-soluble vitamins. Right? So after the break, we're going to present you with a nice piece of bread and butter. Okay? But the bread is of camel wheat, and the butter is unpasteurized. Right? That's when you start to taste what real food should be like, what people ate, 100, 200 years ago. Right? I think that's enough